I want to get, like I said, to some safety and CQA type questions. And I'll start here. Jeff, when we last spoke, I joked around a little bit that no one wants to draw direct comparisons between mRNA vaccine quality and safety profiles and comparisons between that and mRNA therapeutic quality and safety profiles. For obvious reasons, they obviously don't want to talk about it publicly. I'll let our audience read between the lines there on the why behind that. But let's talk a little bit about what regulators are looking for in terms of mRNA therapeutic safety profiles. Do they know yet?
Well, I'm not sure that they completely know yet, right? I think a lot of that really gets down to what is the dose, and which you have to prove anyway, right? You've got to prove that's safe, and then there's going to be repeat dosing for pretty much all of these. What is the frequency that you're going to come back and have to redose? But in the end, like I said earlier, it's the lipids that are the most toxic part. So it's really going to come down to the safety around the lipids. There are definitely some other questions out there: the double-stranded RNA, any immunogenic effects that might occur as well. So there are questions around the mRNA, but I think the biggest questions are going to be around the lipids and how much you can have in there, and is there anything we can do to reduce the toxicity of those lipids? I think that's a big ask, right? That certainly there are some that are less toxic than others, and we'll navigate towards those. But that's going to be one of the big questions, in my opinion. And what they're looking for is more understanding of what your impurities are, both on the lipid side and on the mRNA side. What does an integrity look like? What are the factors affecting that? And what's acceptable to put into humans?
Yeah. On that note, we do have a small trend line in questioning. James and Lynette both have questions about the challenge in dsRNA removal and process development, to the point you just made, Jeff. Any thoughts on that, the challenge and approach that either you can share around dsRNA removal?
I don't have a whole lot that I can share. I mean, we've done a lot of development, and certainly, there are people on my team that are more expert in that than me. If maybe I can take that one back and see if there's anything we can disclose and come back with people about that.
Yeah. All right.
Yeah, I'm kind of in the same position. That's obviously key. That's a key parameter. So there is a lot of time spent in process development to optimize that step. But yeah, there's nothing right now that I would share.
Okay, Dr. Van Cott, I wanted to give you an opportunity if you wanted to address that question around what you anticipate regulators looking for generally in terms of safety profiles.
Yeah, I think Jeff did an excellent job responding to that. I think a little bit more of an unknown. Obviously, we have a lot of safety data, hundreds of millions of doses on mRNA and LNP. I think when we get to therapeutics, you may start to see more work in, circular RNA, self-amplifying RNA, and larger RNA constructs, which, are going to be, different assays that we're going to need to measure. So I think there's going to be, reflection and looking into some of the quality attributes of those new RNA constructs, and so I think that will be the focus as more of those products, with the idea that they can drive higher levels of protein production. I think there's going to be a lot of focus attention on those new exciting constructs as well.
Mm-hmm. It's another good segue to an audience question, Dr. Van Cott. An audience question that sort of challenges the platform approach. So I'm gonna put it in front of you. It's from MR Deep. He wants to know, doesn't a platform formulation, or isn't a platform formulation not feasible for LNP mRNA, considering the constructs do change? So to the point you just made, there are new constructs sort of coming down the pike. So can you, I guess I'd ask either or both of you to rationalize the platform approach in light, in the context of that question.
Yeah, no, I think Amar Deep's question is they're absolutely right. I think what we were trying to find in the platform was you lock in a lot of the parameters, right? So your mRNA design is locked with respect to 5' 3' UTR, your cap, your poly A. Your LNP construct is the same. What you're really just doing is maybe modifying some base pairs within your coding sequence. So like, for example, if it's spike and it's a different strain, or if it's, a string of neoantigens, you're changing sequence, but everything else remains the same. That could be translated into a platform. But you're absolutely right. If you're starting to change your LNP, if you're changing your mRNA design, then you're going to be re-optimizing, and you're going to have to be demonstrating, safety attributes of those products going forward.
Yeah.
Yeah. I mean, if you're changing your mRNA design, then yeah, you're actually working on a new platform, I would argue.
Yeah.
So it's but I think that there's still value if you take that and still say that, okay, well, we have an LNP platform, we have this formulation or whatever that we're trying to fit it in, and we understand it. Even if it's not perfect, it's a great starting point, right? So you didn't gain all of the efficiencies that you would have hoped, but you have a starting point, and then from there you can utilize the information that you previously gained, and then get more information and move a little faster than you would if you were starting from scratch.
Mm-hmm. Mm-hmm. Yeah. What progress have we made in terms of identification to CQAs that result in improved safety and quality? Sort of like, I'm not looking for maybe a chronology or a continuum, but if you kind of had to roll it back the last couple of years, like have there been industry accepted sort of best practices that have moved the needle?
I mean, I think it's a great question. I think
people always say that people always say that's a great question to me when it's like a question that's virtually impossible to answer. That's a great question.
It is virtually impossible to answer. But yeah, I'd say that during COVID, a lot of stuff was published. There was a ton of work done, a lot of stuff published, and I think that really brought an awareness of what was being done to the industry and helped advance the whole area of mRNA and LNPs. So I think just that information, but it also brought up instrumentation, equipment, process understanding. All of that came as a result, and was pretty well shared. I mean, obviously we'd all love more sharing, but there was a lot of information shared, which helped move it forward. So I think the FDA is also, the regulatory bodies are getting more savvy and understand a little bit better. So they're asking good questions as well. And we're having, I think, real conversations about what is needed for these products. Mm-hmm. So it's a combination. It's definitely iterative, and there's a long way to go. But the general understanding that's coming out from the vaccines has been a real plus, even in the therapeutic area.